I'd like to introduce our first speaker, who is Stephen M. Druker. And he is a public interest attorney who initiated a lawsuit against the U.S. Food and Drug Administration that revealed the agency had covered up its own scientists' warnings about the risks of genetically engineered foods and then misrepresented the facts. He has served on the food safety panels at conferences conducted by the National Research Council and the FDA, spoken at numerous universities, including Harvard, Columbia, and Cornell, and met with government officials worldwide, including the heads of food safety for the UK, Canada, France, Ireland, and Australia. Druker received his law degree from UC Berkeley, where he was elected to both the California Law Review and the Order of the COIF, I hope I said that word right, which is the Legal Honor Society. His articles on genetically engineered food have appeared in several respected publications, including the Congressional Quarterly Researcher and the Financial Times. His influential book, Altered Genes, Twisted Truth, How the Venture to Genetically Engineer Our Food Has Subverted Science, Corrupted Government, and Systematically Deceived the Public, was released in March 2015 with a foreword by Jane Goodall, hailing it as, quote, without doubt, one of the most important books of the last 50 years, unquote. Among the other scientists who have praised it is David Schubert, a professor at the Salk Institute for Biological Studies, who has called it, quote, incisive, insightful, and truly outstanding. In 2017, he received the Luxembourg Peace Prize for outstanding promotion of environmental peace. Please welcome Stephen Druker.
Thank you very much. It's a pleasure to be here speaking with you this evening. Now, what do you think would happen if at the snap of my fingers, everybody in the world became instantly aware of all of the facts about genetically engineered food, and everybody's opinion about them became aligned with our best scientific knowledge? Well, in the rendition of, if we adhere to the routine rhetoric of the proponents of these products, we would have to predict that such a worldwide flash of illumination would cause all the opposition to them to vanish like smoke. Because in the rendition of reality that they propound, all of the opposition to genetically engineered foods, or more commonly referred to as GMOs, is based in ignorance of the facts, and all of the concerns about risks are a product of inadequate understanding of the science. But in the world we actually inhabit, the phenomenon that would rapidly vanish is not the opposition to the foods, but the foods themselves. That's right, if the actual facts became widely known, the entire genetically engineered food venture, and I'll be referring to that as the GE food venture, the entire GE food venture would quickly collapse. And that's why, despite their pretensions about wanting to educate the public, the proponents of genetically engineered foods have routinely suppressed and distorted critical information. My book documents case after case in which eminent scientists and scientific institutions have stooped to deception in order to enable the GE food venture to advance. Basic facts of biology have been twisted. The process of creating genetically engineered foods has been deceptively described to make it appear far less disruptive and much more precise than it actually is. And false statements have been consistently issued about the tests on these foods to cover up troubling results. Further, the evidence that demonstrates the distortion of the evidence is solid, and its solidity has been attested by many experts. For instance, John Eichert, a professor of agricultural economics at the University of Missouri, has stated that the evidence presented in my book is comprehensive and irrefutable. Those are his words, comprehensive and irrefutable. And The molecular biologist David Schubert, a professor at the Salk Institute for Biological Studies, has hailed the book as scientifically solid and truly outstanding. Professor Schubert also stated, this is a quote, "It dispels the cloud of misinformation that has misled people into believing that GE foods have been adequately tested and don't entail abnormal risk." Moreover, the irrefutable fact that the facts have been routinely misrepresented is in itself concrete evidence of how strongly the evidence weighs against the safety of the safety of GE foods and the soundness of the venture that's producing them. Because, think about it, if the evidence were truly supportive of safety, there would have been no need to distort it. That should be a no-brainer. If you have the evidence on your side, if the facts support your position, you're proud of them. You boast about them. You present them in their purity. You don't adulterate them. It's only when the facts actually weigh against your position and are quite uncomfortable to you that you have to resort to trickery. And if you do, that's a fact about how weak the facts support you. During the next 40 minutes, I'll point out some of the key distortions and falsehoods and clear up the confusion they've created. In the process, it will become evident that there are strong science-based reasons for reaching 4 conclusions. First, producing new foods through recombinant DNA technology, which is the technical term for genetic engineering, is an inherently risky process, inherently risky. Therefore, 2, every genetically engineered food poses an abnormal level of risk. 3, the safety of those on the market has never been adequately established. And 4th, some of them have already been shown to be harmful. I will also explain how the GE food venture is not only abnormally risky from the standpoint of biological science, but outright reckless when viewed from the standpoint of computer science, and how the biotechnicians have utterly disregarded the hard-learned lessons that computer science has gained about the inherent risks of altering complex information systems. So let's first examine one of the biggest falsehoods that's been perpetrated in defense of GE foods: the routine assertion that there's an overwhelming scientific consensus that they're safe, and that this consensus rests on extensive concrete evidence. Indeed, the strength of this purported consensus is claimed to be on a par with the degree of expert consensus about the reality of human-induced climate change. However, although there truly is a genuine expert consensus in the case of climate change, there has never been one in regard to genetically engineered foods. And whereas every group of experts that has examined the data related to climate change has reached a common conclusion, many well-credentialed experts have raised cautions about GE foods, and several respected scientific institutions have done so as well. For instance, in 2001, the Royal Society of Canada issued a report concluding, A, that the default prediction, and that was their word, default prediction for every GE food should be that the genetic alteration has induced unintended and potentially harmful side effects, and B, that it is scientifically unjustifiable, that's a quote, to regard any GE food as safe unless its safety has been established through a course of testing far more rigorous than any regulators in the world have yet required. That report has never been revised or refuted, and it is as relevant today as it was when it was issued. The British Medical Association and the editors of the Lancet, a premier medical journal, have also expressed concerns about the risks. And the Public Health Association of Australia has recently called for a complete moratorium on the planting and the marketing of genetically engineered foods. The scientific experts at the U.S. Food and Drug Administration have likewise recognized that GE foods entail abnormal risks. This fact came to light in 1998 when my organization, the Alliance for Biointegrity, led a lawsuit that compelled the FDA to hand over more than 44,000 pages of its internal files on GE Foods. Within that trove of documents were a number of memos from the agency's own scientists expressing concerns about the unusual risks of genetic engineering and the need for all the foods it produces to undergo rigorous safety testing that's capable of detecting the potential unexpected side effects that could be reasonably expected. The pervasiveness of the concerns is attested by an FDA official who studied the expert input and declared, quote, "The processes of genetic engineering and traditional breeding are different, and according to the technical experts in the agency, they lead to different risks." Now, please note that that official did not say there's some debate going on among our scientists, and some of them have concerns. She stated, according to the technical experts in the agency, these foods lead to different risks. An unequivocal statement because, as the memos show, that was the overwhelming consensus within the FDA's own scientific branch. There wasn't a question about whether they were risky. It was that they did entail unique risks, that genetic engineering is different than traditional breeding. Moreover, the FDA's biotechnology coordinator acknowledged there was not a consensus about safety in the scientific community outside the agency either. However, the FDA has an admitted agenda. It's in writing. They've admitted this in writing, that they have an agenda to foster, quote, to foster, those are their words, biotechnology. And when the agency issued its policy in May of 1992, it claimed that it was not aware of any information showing that foods derived by genetic engineering differ in any uniform or meaningful way from other foods. Yeah, I don't believe it either. I mean, their files are chock full of memos from their own scientists explaining why these foods entail different risks, why they are different. The FDA also asserted there was an overwhelming consensus among scientists that GE foods are so safe they don't require any testing, even though it full well knew that no consensus existed. Accordingly, the FDA has allowed GE foods to enter the market without requiring even a smidgen of testing. That's something most people don't understand. No testing is required by the FDA. They make some show of some review process. It's all a sham. When you cut through all the hyperbole, there is no requirement for testing. In fact, the FDA doesn't even require that a manufacturer of genetically engineered foods gives it advance notice or any notice subsequent to the dumping of GMOs on our market. So the FDA actually has admitted it doesn't actually know for sure all of the GMOs that have entered the US market because it doesn't even require that it be given formal notice. That's how loose it is. Now, if the FDA had told the truth back in 1992 and disclosed the extensive concerns of its own experts, the subsequent history of the GE food venture would have been very different and most likely very short. At the least, any GE foods that did reach the market would have been subjected to much more rigorous testing than regulators anywhere have required. So that's one claim discredited. What about another one, another major one? The oft-repeated claim that no ingestible product of genetic engineering has ever been linked to a human health problem. That's baloney too. In fact, the technology's very first ingestible product caused a major epidemic here in the United States. Dozens of Americans were killed, and between 4,000 and 5,000, according to the Center for Disease Control's most recent estimate, were seriously sickened. Hundreds of those individuals are still invalids. They're still incapacitated in one significant way or another. That epidemic occurred in 1989 and 1990. Most public health officials with whom I've talked aren't even aware that it happened. I'm getting ahead of myself here. So what was the cause? What was that first ingestible product? The product was a food supplement of the essential amino acid L-tryptophan. We all need it. It's just some people who are under a lot of stress, are having difficulty sleeping, or are having a lot of nervousness, needed more than they were getting from their average diet. So hundreds of thousands, probably millions of people around the world have been taking L-tryptophan supplements on a fairly regular basis, and it helped them. It was a safe, natural alternative to, to pharmaceutical drugs, or to having to eat a whole lot of turkey to get your L-tryptophan that way. For years and years, no problems had ever been associated with taking supplements of L-tryptophan. But then something unusual happened. One of the main manufacturers, the main manufacturers were all in Japan, one of the main manufacturers got the idea that if they genetically engineered the bacteria in a certain way to produce more L-tryptophan in the same amount of time, they could make more money. And so they did. But they didn't really explain to anybody that's what they were doing. It turned out it was their only the supplements manufactured by that corporation have been linked to the problem. And now, and this is very important, although the L-tryptophan supplements that were derived from genetically engineered bacteria met the standards for pharmacological purity. They tested pure pharmacologically. Like all other tryptophan supplements, it contained minute impurities, minute level of impurities. You can't get rid of all of them. But at that level, at that minuscule level, they shouldn't have hurt anybody. In fact, the other tryptophan supplements had minute levels of impurities too. They weren't hurting anybody. But at least one of the accidental additions to the genetically engineered L-tryptophan was highly toxic at an extremely low level and caused that epidemic. Consequently, excuse me, got ahead of myself. Now, because of none of the tryptophan supplements that were produced via the non-engineered bacteria had ever been linked to disease, and because genetic engineering can create unintended disruptions within the altered organisms, there were legitimate reasons to suspect that the engineering process had induced the formation of the extraordinarily toxic substance or substances that caused the calamity. Consequently, the proponents of genetic engineering strove to convince the public that the technology was blameless. And to do so, they issued a string of deceptive statements. Those deceptions have been so successful that despite the fact that the evidence points to genetic engineering as the most likely cause of the toxic contamination, most people who know of this tragedy are under the illusion that the technology has been exonerated. Worse, because GE proponents routinely claim that none of its products has ever been linked to a health problem, most people aren't even aware that such a catastrophe even happened. As I mentioned, even most of the public health professionals with whom I've spoken, and I've spoken with a lot, were not even aware that there was an epidemic that was linked to a product of genetic engineering, let alone the very first one that came to market. Now, it's important to note that the toxic tryptophan incident has serious implications for all foods produced through genetic engineering. Why? Those bacteria were not altered with any foreign genes. They were simply endowed with extra copies of some of their own genes that are involved in the synthesis of L-tryptophan. And they were not engineered to produce anything other than L-tryptophan, which is a beneficial substance they ordinarily make. But it was the forced overproduction of this normally benign substance that apparently put abnormal stress, okay? Normal substance, abnormal overproduction apparently led to abnormal stress And that in turn led, apparently, most likely according to the evidence, to the creation of an unintended and highly toxic byproduct. Now, almost every genetically engineered food-yielding organism is being compelled to overproduce one or another chemical, sometimes more than one. Many of these new so-called stacked GMOs are overproducing several different foreign proteins. So for that reason alone, just about every genetically engineered food entails an abnormal risk because it's overproducing a protein, usually a protein, maybe another substance, and that in itself is an unnatural and stressful situation from the standpoint of the organism itself, which was never consulted about, never volunteered to undergo that alteration, be subjected to that stress. It's additionally important to keep in mind that the lethal tryptophan supplements had passed through a filtration process and yet were still toxic, while genetically engineered fruits, grains, and vegetables do not undergo filtration at all. Okay, let's examine another claim. The standard claim, I do want to mention an entire chapter of my book is devoted to that L-tryptophan incident. It's very extensive. It has been reviewed by many eminent experts, and it has been strongly praised by, among other experts, but especially by a molecular biologist who for 10 years was a professor at the Mayo Clinic, and he led a team that actually was researching the L-tryptophan incident. He and his team probably know more about it than any other researchers, and he praised my chapter as the most comprehensive, balanced, and accurate account of that catastrophe he's ever read. And he put that in a sworn affidavit, just so people, it was up there. Yes?
Let me make a comment. I really appreciate hearing this.
Well, can we save your question till the,
Yeah.
I'll get to you. We can call on you first.
I'm just wondering if you could ask a show of hands of how many people knew that GE was involved with the L-tryptophan event when it happened.
Okay, how many who knew it? Not very many. And this is one of the most highly educated groups in the country and the world about health and GMOs, and just one person knew it ahead of time. That's pretty astounding. Okay, so it was a good question to do it at that point. And all right, so that in itself tells you something. How, in fact, that's why the title of that chapter is titled Disappearing a Disaster. And the subtitle is How the Facts About a Deadly Epidemic Caused by a Product of Genetic Engineering Have Been Systematically Obfuscated. And when you read the chapter, it's astounding to see how they've done that. I mean, one deception after another, and they've misled even a blue ribbon commission in New Zealand that was supposed to find the facts and report to the government. It has several just false statements about that L-tryptophan incident, and then concluding that it was completely exonerated. Here's an interesting thing. They stated that there were lawsuits in the United States, and the United States court system had definitively determined that the toxic contamination had not been caused by genetic engineering at all, but was caused by some other aspect of the manufacturing process. I contacted the main lawyer who handled all of the litigation. He defended the manufacturer and all of the litigation. First, in almost every case, the cases were settled out of court. Nothing was determined. There were only a couple cases that even went to trial. But because our liability law is strict liability, all that the plaintiffs had to prove was that that L-tryptophan supplement had caused their illness. There was never an issue as to what aspect of the manufacturing process caused the contamination. It was never at all an issue. It was never decided. He was amazed that a Blue Ribbon Royal Commission could have stated that. Where did they even get that? Some of that propaganda deception. But that's how bad it gets.
Didn't the FDA ban the L-tryptophan then?
I'm sorry, what?
Didn't they ban?
Yeah,
supplements?
let's follow up during they did. And we'll go into it in more detail because I want to make sure I get, but if you want to go more into that, we can. Okay, now let's move to the standard claim that the safety of genetically engineered foods has been thoroughly established by reliable testing. Well, that claim cannot survive scrutiny either, especially considering that many well-conducted studies published in peer-reviewed journals have detected harm to the animals that consume genetically engineered food. In fact, in 2009, a systematic review of the toxicological studies in the peer-reviewed literature on genetically engineered food that was itself published in a peer-reviewed scientific journal concluded that the results of most of them indicate that the products, quote, may cause hepatic, pancreatic, renal, and reproductive effects and may alter hematological, biochemical, and immunologic parameters, the significance of which remains unknown, unquote. Another review that encompassed the additional studies that had been published up until August of 2010 also provided cause for caution. It concluded there was an equilibrium between the research groups suggesting that GE crops are as safe as their non-GE counterparts and those still raising still serious concerns. That last part, those raising still serious concerns, is a quote from the researchers. So obviously, the fact that more than 15 years after GE foods had entered the market, half the published studies on them raised serious concerns in the eyes of objective scientific reviewers undermines the claim that their safety had been or has been scientifically established. And this conclusion is fortified when we examine some of the specific results that accrued both before and after that date. For instance, a team of university scientists published a paper in 2011 in which they reviewed the data from 19 of the feeding studies on GE soy and corn varieties that had already gone through the regulatory process, were being eaten by people here in US and Canada and several other countries, and they comprised 83% of the GE foods that the people have actually been eating. Okay, so 83% of them. Well, what they found was disturbing. Now remember, these data, the data they reviewed had already gone through the review process in the EU and Australia. In the US, it just sailed through with a rubber stamp. Canada went through there too. They found 9% of the measured parameters, including blood and urine biochemistry and organ weights, were significantly disrupted in the animals that had eaten the GE feed. Somehow the regulators hadn't noticed that. Moreover, the greatest disturbances were to the kidneys of the males and the livers of the females. And the scientists emphasized that because livers and kidneys are the main reactive organs in cases of chronic food toxicity, those findings should have been viewed as danger signs, something the regulators had not seen fit to do. Furthermore, when negative results are so disturbing that they cannot be ignored, they're vehemently and unjustly attacked, and the research is routinely misrepresented. A prominent role in this misinformation campaign has been played by the UK's Royal Society, the world's oldest and most prestigious scientific institution. For example, they've been a main promoter of genetically engineered food, and they've actually besmirched their reputation in trying to uphold them and trying to defend them. For example, the Society recently declared that no research has indicated that the genetic engineering process itself has caused any harm, and that all problems that have been detected have been attributed either to the specific gene introduced or to particular agricultural practices that were involved. But that assertion is flat out false. One major study did specifically link the GE process with harm, and it was published in the eminent journal The Lancet in 1999. And it revealed that GE potatoes producing a foreign protein that's safe for mammals to eat caused a problematic effect in the rats that consumed them compared to the rats that ate the non-GE counterpart potatoes that had been spiked with the same level of foreign protein that was synthesized within the genetically engineered potatoes. So therefore, it was very well controlled. to find out whether the foreign protein was going to be doing something or it was something other than the foreign protein. Foreign protein was ruled out. So that's why the researchers, and it was conducted by some of the most preeminent food safety experts in the world, and they concluded that there was something then in the genetic engineering process itself that had caused the results that they detected. Of course, that was viciously attacked. In fact, the two main researchers were sacked, fired from the Rowan Institute in Scotland, even though the British government had actually funded the study. Once it came out with bad results, they shut it down, destroyed all of the genetically engineered potatoes, so nobody could actually do follow-up studies. That's not science. They should have retested it to see if they didn't like the results, they should test them and test those potatoes again and see if they got the same results. But they completely closed down any chance to actually follow up on that study, destroyed all the potatoes, fired the main researchers, husband and wife team, destroyed the reputation, stressed him so badly he got at least one heart attack trying to defend his reputation. That's what happens. And the Royal Society was at the forefront of those attacks. Now, just a few days ago, an article I wrote that goes through, actually chronicles or documents the Royal Society's multiple misrepresentations in defense of GM foods, genetically engineered foods, and explains and shows how they're all wrong, was published on the website of Independent Science News. Maybe some of you have read articles there. They're very careful. It's edited by molecular geneticists, and they scrutinize what is submitted. So if you want to read that article, I think they called it UK Royal Society, a case study in the systematic misrepresentation of genetically engineered foods. And again, the website is Independent Science News. It is only through the systematic misrepresentation of the facts by respected institutions and individuals and their willingness to disregard the ominous implications of the evidence that the GE food venture has been able to continue. And this disgraceful activity is being carried out in the name of science when it is actually subverting the precious principles of science. The extent to which the GE food venture has failed to be evidence-based and instead has rested on the denial and the disregard of the evidence has been vividly summarized by Dr. Michael Antonio, a molecular geneticist at King's College London School of Medicine. And before reading Dr. Antonio's quote, I want to emphasize he is not a foe of genetic engineering in general. In fact, he employs genetic engineering, recombinant DNA technology, in his medical work. Okay? He's using it to try to understand and cure human genetic diseases. So he understands how powerful and uncontrollable in several ways this technology is, whereas he feels that it does have some legitimate applications in the health field. He feels it's unjustified to be, and scientifically really unsound, to be applying it to create new varieties of food. And he's an expert, he's studied the research, he's actually co-authored an important book, GMO Myths and Truths, and here's what he stated. If the kind of detrimental effects seen in animals fed GE food were observed in a clinical setting, the product's use would be halted and further research instigated to determine the cause and to find solutions. However, what repeatedly happens in the case of GE food is that despite increasing evidence of serious adverse health test results, government and industry continue unabated with the development, endorsement, and marketing of these foods as if nothing has happened, to the point they even seem to ignore the results of their own research. Pretty powerful statement, and he knows what he's talking about.
What's his name again? Name?
Back the slide up, please.
Michael Antonio.
He was asking, what is his name? What was the doctor's name?
Oh, Michael Antonio.
Thank you.
I'll go back and show the slide. Whoops, I'm going forward, sorry. That's stepped ahead. That was a preview of coming attractions. Michael Antonio,
Okay.
okay, a very brilliant scientist. and he's got a lot of integrity. So even this short summary has revealed that from the standpoint of biological science, that GE food venture is significantly unsound. And when it's analyzed from the standpoint of computer science, the picture becomes even more troubling. Now, such an analysis is highly relevant because both genetic engineering and computer science are engaged in altering complex information systems. And computer science has learned a lot about the risks of making such alterations. Moreover, it has learned that these risks are inescapable. Inescapable. Software engineers have learned, by the way, Randy, who introduced me, is a software engineer, and at dinner I invited him to scrutinize what I'm saying carefully and to challenge me if he thinks I've gone astray anywhere. I don't think I have, because the chapter in my book on this analysis and what I'm sharing with you has been vetted by several experts. But you never know. Randy may see something that has slipped by others. Software engineers have learned that when the information systems they themselves create become large and complex, there's no way to alter them with complete precision. Even when the alteration is a small refinement designed to improve the system's performance, the mere process of revising it in such an ostensibly minuscule manner is likely to disrupt one or more of its other components. This is an amazing phenomenon. Software systems are designed to be linear, which means they're structured so that a specific operation only produces a specific result. Operation X should only produce Y. However, despite the programmer's best efforts, their systems transcend the intended limits and, to a significant degree, behave in a nonlinear manner. There's a very high likelihood that some of the parts will interact in ways that were not planned and cannot be predicted, which means that Operation X will not only yield Y, but might also generate Q and Z. Consequently, to reduce the potential for unintended interactions, software designers separate components that shouldn't interact and try to insulate them from such interaction. What they try to avoid is creating code that resembles this. They try to avoid writing what they refer to as spaghetti code, and they use that term because in a mound of spaghetti, think of it, it's very difficult to even see where one component goes, and you can see the components are intertwined, and it's almost impossible to kind of work on one without creating a lot of slithering going on in the other ones. So they want to avoid spaghetti code. What they want instead to create is code that looks like this, which they call ravioli code. So they aim to create software programs in which the components that should not interact with one another are as separated as the packets of cheese and vegetables in separate packets of pasta on a plate of ravioli. However, even though programmers have succeeded in designing systems that are far more analogous to a plate of ravioli than a mound of spaghetti, and have reduced the potential for unintended interactions, they have not been able to eliminate it. Such unwanted results do continue to happen, just at a much more reduced rate than in the earlier era of software engineering. Before examining how these risks are dealt with by the software engineers, let's compare the characteristics of human-designed information systems with those of bioinformation systems. Let's compare man-made software with nature's software. As I noted, human-made systems are designed to be linear, and although they unavoidably become nonlinear to some degree, they for the most part function as linear systems. But the situation is very different in the case of bioinformation systems. They are inherently nonlinear. The various parts are intricately interconnected, and every action can create a wide range of effects, many of which cannot be predicted. So again, we have in the human-designed information systems designed to be linear, function in a largely linear manner. Bioinformation systems inherently nonlinear, and they function in a highly nonlinear manner. Moreover, in their endeavor to maximize manageability, software engineers avoid creating spaghetti code. But bioinformation systems are the most extreme instances of spaghetti code. And even if a human being would be able to create them, he or she could not adequately comprehend their various interactions. So human-designed systems, spaghetti code is avoided, minimization of interaction between different components. Bioinformation systems, the ultimate spaghetti code, unfathomable degree of interaction between the parts. Moreover, despite the substantial knowledge that we humans have gained about the workings of bioinformation systems, the extent to which our understanding remains deficient should be profoundly humbling. In human-designed systems, the rules governing how the parts interact are clearly expressed in written form, but only a small fraction of the rules of bioinformation systems are known, and most of those pertain to the mechanics of gene expression. Yet, as numerous experts have emphasized, many of the most important rules do not operate at the level of the genes. For instance, Richard Stroman, who was a distinguished professor of cell biology at the University of California, Berkeley, and was one of the scientist plaintiffs in the lawsuit I organized, a really wonderful man, a lot of integrity. He passed away a few years ago. So Dr. Stroman asserted that the most important rules operate at a higher level, the level at which genes are organized into what he called functional arrays. And he noted that this level of gene management is not confined within the DNA, but is, quote, coextensive with the cell itself. Moreover, he emphasized that the dynamics operating at this level are different than the dynamics operating at the lower levels, and that the interactions are far more complex to the extent they are ultimately, quote, transcalculational, which, as he noted, is a mathematical term for mind-boggling. Several other experts have also pointed out that bioinformation systems transcend DNA and ultimately extend throughout the entire organism, while also extending far beyond our comprehension. So again, human-designed information systems, mind-made; bioinformation systems, mind-boggling. Okay? On the one hand, the system's rules and dynamics are comprehended by those who make the revisions. On the other hand, they're transcalculational. Revisions are made with very scant comprehension. Now, in addition to the vast differences in the degree to which humans understand how the two types of systems operate, there are also glaring differences in how they make revisions to them. Software engineers insert new code precisely where they want to, without accidentally disrupting the way in which other code is written. Further, no unintended code enters the system. In contrast, bioengineers have been inserting DNA haphazardly. Their insertions have invariably disrupted sections of native DNA, and unintended pieces of DNA have almost always entered as well. So, revisions are precise and minimally disruptive on the level of human-designed information systems, but with bioinformation systems, the revisions through genetic engineering are imprecise and highly disruptive. And that's well documented in scientific research, the levels of disruption, the alterations in gene expression, and hundreds of other genes. So in light of these enormous differences, it would be reasonable to think that genetic engineers would exercise far greater caution than do software engineers. But in reality, they exercise far less. Software engineers recognize the inescapable risks of altering complex information systems, and they deal with them responsibly. Accordingly, when they revise programs, they routinely conduct tests to detect whether any problems have been created. Moreover, there's a special class of software for which the testing is extremely rigorous. It's called life-critical software, a class of software that if it fails, human life also will fail. People will die or be seriously injured on a broad scale. Now, examples of such life-critical software are the programs that govern pacemakers or X-ray therapy machines, or in the case of aviation, airplane guidance systems. Okay? You don't want bugs in those systems. In the United States and the European Union, not only are such programs required to undergo strict testing before they're even allowed on the market, they also must be rigorously tested anytime they're revised, no matter how minor the revision. The regulators will not accept arguments that the revised program is substantially equivalent to the former version. There must be a demonstration of safety achieved via systematic, stringent testing. But there's a glaring contrast in how risks are handled when disruptive alterations are made to the biggest, most complex, and yet least understood information systems on our planet. The biotechnicians don't adequately acknowledge the risks, and they largely deny them. That's very important. They are in denial. My book documents case after case. They deny that there are even any risks. They deny problems. Even the regulators have discounted the risks and assume there's little need for rigorous testing. As I mentioned before, the US Food and Drug Administration claims that GE foods are so safe they don't have to be tested at all. And although regulators in the EU and most other regions have required some testing, it's been minimal. For almost 20 years, genetically engineered foods were allowed on worldwide markets based solely on some superficial showing of substantial equivalence to their conventional counterparts, something that is never allowed in the case of life-critical software, despite the fact that a toxic tomato could cause many more deaths than a malfunctioning X-ray machine. And although the European Union has finally, just a few years ago, required safety testing with a whole food, the tests are still remarkably lax by software standards. Even if GE Foods were compelled to undergo the same testing that's mandated for new pharmaceutical drugs, it would still fall short of the level of rigor that's required when changes are made to life-critical software programs. So, human design information systems, small revisions to life-critical systems are presumed to entail substantial risk, and rigorous testing is required. With bioinformation systems, major revisions to life-critical systems are presumed to be essentially safe. There are lax requirements and lax testing. A very crazy situation. Moreover, as my book demonstrates, if the kind of rigor that's required in software testing were mandated for GE Foods, the entire venture would implode. Consequently, it is incurably risky. There's no way, and that's because there are several reasons. One of them is it's very expensive to do the kinds of testing that's conducted on pharmaceutical drugs. And one reason pharmaceutical companies claim that they should be able to continue charging monopoly profits, high profits, for until the patent expires and the generic competitors come out, is they say we have to recoup all of the expense of the research we do. Well, you can charge a premium for a pharmaceutical drug. There's only so much you can charge for soybeans and rutabagas and sugar beets. So there's a natural limit on how much you're going to be able to charge. So as the testing becomes more expensive, the project becomes economically less and less feasible. So in order to get scientific reliability through the level of testing that would be needed, you can't really have the scientific reliability and the economic viability. Okay, that's an important point. There's no way in which the genetically engineered food venture can achieve both scientific reliability and economic viability. Scientific reliability, did I say that? There cannot be the scientific reliability and economic viability. That's why they rigorously fight any efforts to impose more testing. And of course, there's another reason, psychological reason. The more the public understands there are risks, the less they're going to want these foods. In fact, there would be a huge wave of revulsion and righteous indignation if people understood how they've been hoodwinked and how they and their children have been subjected since 1995 to foods that the FDA's own experts stated entail abnormal risks. It's just astounding. Now, you may be wondering, I often scratch my head, how is it that the people promoting genetic engineering could have remained so oblivious to the risks of altering complex information systems? Well, a big part of the answer is quite simple. They didn't even understand, they didn't appreciate that they were dealing with complex, intricate information systems. Back, as strange as it may seem to us now, based on the information that has come out about the complexities and the intricacies of the genome, back in the 1970s, when genetic engineering first became possible, the understanding of the genome was very primitive and mostly wrong in many key ways. And so back then, scientists, molecular biologists, presume that genes act independently, that they're kind of independent agents, that they are not arranged in an organized manner, and that the sequence in which they occur is essentially unimportant. Accordingly, they also presumed that the genome is a simple linear system in which the action of a single gene will not significantly impact the others and won't disrupt their normal function. And for instance, in a 2007 New York Times article, the veteran technology reporter Denise Caruso observed, "The presumption that genes operate independently has been institutionalized since 1976, when the first biotech company was founded. In fact, it is the economic and regulatory foundation on which the entire biotechnology industry is built." This set of presumptions that I've just described supported the belief that a chunk of recombinant DNA could be wedged into a plant's genome without inducing disturbance, because if the behavior of the native genes was largely uncoordinated and their arrangement was irrelevant, there would be no important patterns that could be perturbed. Accordingly, they engendered confidence in the precision of genetic engineering by implying that its results would be consistently predictable. That was the belief and the claim. Oh, this is precise and predictable. We know what's going on. We can predict all the consequences. However, these influential assumptions, which provided the ideological foundation for a colossal venture to transform agriculture, have been decisively discredited. Abundant evidence has demonstrated that there is a high degree of coordination between genes and that their arrangement is highly organized. And this evidence undermines the claims about the safety of genetic engineering. Thus, in her previously mentioned New York Times article, Denise Caruso asserted, "Evidence of a networked genome shatters the scientific basis for virtually every official risk assessment of today's commercial biotech products, from genetically engineered crops to pharmaceuticals." Now, I have to point out that Caruso's, as you notice, Caruso's article came out in 2007, 10 years ago. It still doesn't seem to have had any dent in the thinking, any impact in the thinking of the promoters of genetically engineered foods. And that occurred in the New York Times. So it's ignored. People do not come to terms with it. Secondly, Denise Caruso, to the best of my knowledge, I've read a book of hers, I've read articles, she wasn't even taking into account the abundant information that's been derived from software engineering that I've just shared with you. So even without taking that into account, just based on the genetic, the biological science perspective, she states that the scientific basis for the whole GMO project has been undermined. So when we factor in all of the abundant information from computer science, then we can see how it really has been undermined, how this is extremely dangerous, and in fact, that the genetically engineered food venture is not just risky, but as I said before, outright reckless the way it's being done. Of course, I think many of you have heard within the last few years of a new set of techniques for altering genomes that are referred to as gene editing. And you know, it's interesting, they keep saying, now these are precise. Well, for years we kept hearing that recombinant DNA technologies, they've been doing it is very precise. Somehow now, well, that was precise, but this is ultra precise. So it wasn't all that precise actually. But, and of course, CRISPR-Cas9 is one of the most important of those and has great promise and great potential for harm, it appears, in the, in the human health field and in genome editing of the human genome or animal genomes, but the genetic engineers want to apply that to food production as well, and they claim it's going to be very precise and safe. But is it? Not from a standpoint of computer science. For instance, those techniques can still induce accidental off-target effects. That's still the case. But even if they didn't, as is the case with previous modes of genetic engineering, in order to transform the altered plant cells into seed-bearing plants, tissue culture must be employed. Now, this is very important. Even with CRISPR, they're not genetically engineering, they're not going to be genetically engineering seeds that you can then plant. Maybe they'll be able to do it at some point, but up to now, cells from adult plants are being taken and they are being altered. But once you've done the genetic alteration, you can't take that adult cell and put it in the ground and expect it to grow into anything. It'll just die. So in order to get those cells basically coaxed, and it's more likely to say forced, more apt to say forced into mature plants, a very artificial process is employed, which, as I said, is called tissue culture. And plant hormones have to be applied, and then nutrients. And when a plant develops through tissue culture into an adult, it goes through a very significantly different process of development than when a seed naturally grows into a plant. And it's known, tissue culture is known to create many mutations throughout the genome. It's highly mutagenic. And in fact, some scientists have dubbed the kind of changes that it causes genomic shock. So, there goes the predictability and precision out the window if it goes through the tissue culture process. But again, the genetic engineers and their defenders don't really talk about tissue culture. They don't want to get into all that because it would give the lie to their claims of precision. Let's do it during a discussion. I'm almost done here. So let me just, genome editing, you can see CRISPR written out if you didn't get it. CRISPR-Cas9, not a precise procedure. From the standpoint of computer science, such techniques entail abnormal risks and must be rigorously tested. They certainly are creating greater potential for unintended effects than are precise revisions to life made by software engineers to life-critical software programs. So they need to be carefully tested. By the way, there's a huge propaganda campaign underway now in the European Union and elsewhere to state that anything created, any GMO crops created through CRISPR shouldn't even have to be tested. They should just sail through because, hey, it's precise editing, we can predict it, no problem. It's just not the case. And that's why this analysis from the standpoint of computer science is so important. It's an extremely important branch of science. It has been almost utterly disregarded, and it shouldn't have been. I'll just mention, because it is surprising to me, I'm a lawyer, although I've majored in philosophy at Berkeley, studied a lot of science. Within the first few months that I started studying genetic engineering, wanted to learn about it, I realized I need to interview software engineers, and I know several, because they are altering, they're creating and altering information systems. I want to know what they and their discipline know about it. And it was amazing, eye-opening to start learning about it. And they were shocked when they actually realized what was going on in the standpoint from the genetic engineers. I don't think I have yet talked with a software engineer who wasn't aghast when they actually understood what was going on with such limited testing. It's pretty astounding from the standpoint of software engineering. So if we cut through the promotional propaganda that's been passed off as science, and we carefully examine genetic engineering, in the light of our best scientific knowledge, knowledge gleaned not only from biological science, but from computer science, it becomes clear that the venture is incurably risky and that the risks are unacceptable, especially when safe, sustainable, and well-proven alternatives are readily available. Thank you very much for your attention.
Okay, so I think, do you want to ask the first question?
Yeah, this deals with what you were just talking about 10 minutes ago, about this precision about putting these genetic modifications in a certain place in the chromosome. And what comes to mind is this term that I've just got in my head at the moment called gene migration, which would represent the fact that what you put there may not stay there. It may replicate itself when we've got RNA in the RNA process. It may find itself in other locations.
Right.
I mean, that just came into my mind. I don't know if that is a problem or not, but do you have any way of
Now,
addressing
are you
that?
a molecular biologist? Okay.
No, I'm
If
just
there one, I think it might be a problem.
I'm an anomaly. I'm a citizen, but
It could be, there's so many other problems, that's one we don't need to look at, but there are multiple ways in which genetically engineered foods entail risks that are not present in conventional foods. That could be one of them. We know genes can move around. Of course, there's a class called transposons, or commonly called jumping genes, that we know do.
Right.
And now it's interesting, the proponents of genetic engineering, and this includes some esteemed molecular biologists, claim that, oh, well, they're jumping genes in conventional foods anyway, and so what we're doing isn't any different. Well, actually, most so-called jumping genes don't jump around very much, and their positions have been there for a long, long time, in biological time. But it's known that the genetic engineering process, one of the disruptions that it can create is to stimulate jumping genes into jumping. So to the extent that there is a risk from jumping genes, genetic engineering actually increases that risk in the products that it's being used to change. And again, that's something that they do not deal with either. So that's just one of the problems. Now, hopefully your question was answered because the process itself is risky. So that's why I wasn't even focusing on the foreign protein. And usually the proponents try to get everybody focused on the particular protein that's going to be the intended expression product, and then saying, well, this is safe. And again, even their tests on that are shoddy. I'm going to shut that down. I better shut that down now too, because I didn't. So, well, anyway.
We have one more question over here.
I didn't mean to do that. I hit something and got out of the program, and I don't want to have you have to see all that, so I'll just shut it down. Okay, so shows how even this simple technology sometimes doesn't work. Okay.
The question came up earlier about.
I wanted to finish my point.
Okay.
So the attention, they try to put so much attention on whether the known expression product is going to be safe, and even those tests are actually not reliable. And we can go into that more if you want to. And they try to ignore the unintended, the background unintended problems, which are inherent in the process itself. So again, it's a classic case of shifting misdirection of attention. Okay.
Yeah, I'm quite familiar with the tryptophan EMS fiasco. And by the way, the FDA never really did ban tryptophan.
Well, not
They
completely.
merely told all the reporters and everybody in courts and stuff that they had banned it, but they never actually did. They never went through any paperwork to do that. Okay.
But it was off the market over the counter for adults
That's
for a long
right, but
time.
they blamed it on all tryptophan.
Yeah, I know.
So that's
My book goes into that, and they did it.
Yeah.
They, as you probably know, years before that epidemic, the FDA had been trying to move against L-tryptophan and other supplements, especially L-tryptophan, because it was a major threat, to some of the pharmaceuticals. Yeah, because it was a cheap, natural alternative, and it worked.
Yeah.
And they had been trying to get it off the market for years, and they had been fought in court, and they didn't succeed, and that epidemic gave them the excuse to do it. My book goes into that. Okay.
Yeah, but when one marketer in Arizona who was bypassing the law by selling tryptophan,
interesting.
human pharmaceutical tryptophan for veterinary applications, for pets, he went to the FDA and forced them to admit that they had never banned it.
Oh, interesting. Well, I learned something new.
Yeah.
Thank
The other
you.
thing that I'd like to point out is that there's an entirely different level of complexity in terms of proteins. The protein folding problem means that if you take a string of information that would be coding for amino acids, and you take that same string and you put it, let's say you reverse it. So instead of it going A, B, C, D, E, F down to Z, it goes Z, Y, all the way down to A. It doesn't come out with the same protein, even though it's exactly the same sequence. If you change the electrolyte balance, you change the pH, you change the temperature, the protein folds in a different way.
Right.
And we know from Creutzfeldt-Jakob syndrome and mad cow disease that a misfolding protein can be catastrophic to the
Very
health
good
and
point.
welfare of an individual.
And
so if you take a protein that's found in one plant or in an animal and you put it into a potato, if the electrolyte balance and the pH and all those conditions aren't identical, it won't fold into the same protein that's
such an
present in
excellent
the
point.
original food.
My book discusses that too. Are you you're a health professional, or you know a lot? I just wonder what your degree is.
My degree is in organic chemistry.
Yeah, okay, good. And your point was beautifully made, and my book goes into that as well. And there's evidence of protein misfolding as well. And, especially, it can be, here's the thing, besides protein misfolding, and I'll just make this point too, when a gene from a bacteria is put into a plant, there can be another level of problems, and that is because plants often make additions to proteins, often sugar chains or other molecules. Bacteria don't do that. So plants are probably, in most cases, making additions to the protein that's synthesized, even if it is correctly folded. Then it has additions, maybe sugar chains, that weren't there in the bacterial version. They could make it harmful, allergenic or toxic or otherwise harmful. So again, they're messing around with something very, very complex and treating it again as if they're making some simple mechanical linear system, which it's not that way at all. It's really an insult to nature how they describe it and how they're handling it. Yes.
Hi, my name's Kiki, and I'm a naturopath, and I'm the Nevada labeled GMO leader, started that organization. And we've used your book. I want to thank you so much for your years of work with our city councils to get some pesticides. They're in the process of getting bans created, and your books really helped with that tremendously, because they don't believe you when you go in and tell them the facts, right? But anyway, I have a question for you as an attorney. I just read an article. There's a new GE product that came out that's a vegan burger.
Oh, yeah.
Have you
I haven't
heard?
been able to keep up to date
It's
because
here
I'm
in Silicon
traveling.
Valley, the home of that company. I don't know the name. And it's an all GE product. And a man went into anaphylactic shock, one of the first people who ate it publicly. And of course, they're denying that it had anything to do with it being GE. They have no...
Well, they always will.
Yeah, they have no approval from anybody that... They were actually told not to produce it, and they're producing it. I'm shocked, as a citizen, not just as a doctor, but as a citizen, I'm shocked that a government agency can tell a corporation this has not been, there's been no research on it.
Yeah.
One person already has become almost died. And where do we have power? I mean, you had a lawsuit forever and couldn't complete it. What can we do? Because this is ridiculous.
It is.
Yeah.
It's more than ridiculous, but it is. So the question is, what can we do? By the way, that new food, as I said, they often say this food has gone through approval. From the FDA side, nothing has to get approved. As I said, they don't even have to tell the FDA. So just understand, from the standpoint of food safety, if the FDA is involved, somebody can just bring it out and put it on the market. Now, it is illegal, technically, but the FDA, as far as the FDA is concerned, it is legal. The FDA is letting them do it because it's breaking the law. It's actually an astounding situation. But what can we do? Well, there are many things you can do. First, try to protect yourself and become more rigorous in avoiding GMOs and getting your friends to, because there is the power of the purse. You vote every time you go to a store. And if you avoid foods with GMOs, and especially if you tell the store manager or you're eating in a restaurant, you say, why are you cooking with canola oil that's, it's GMO, I don't want to eat it. Use sunflower oil. If they hear that from enough people, and you say, I'm not going to eat here anymore unless you switch, if enough people tell them, they're going to switch. It doesn't take much shift in consumer habit because the food industry, it's a very low profit margin. So it doesn't take much of a shift to have a food company wake up and think, uh-oh, people are not buying this enough, and if they know why, then they're going to make a shift. And it's already happening a lot. You'll see even Campbell's, I think, is now labeling if they have GMOs, and more and more manufacturers are getting that non-GMO project verification on their product, and it's the non-GMO, organic and non-GMO are very, very hot brands, hot classification. So the more that you and the people you can influence keep shunning foods that you know are GMO, and that, by the way, is almost any product that has derivatives from corn, soy, canola, sugar if it's not labeled cane sugar, because that'll be from GMO sugar beet, papaya if it's from Hawaii or Thailand. That takes into account a lot of them. They are labeled. If they say they have soy and they're not non-GMO verified or not certified organic, you can presume that they're GMO. So in that sense, they're labeled. You just avoid them. And if enough people do that, and more people are, it is making a shift right now. That's a huge shift. I don't see any hope through the U.S. Congress right now. As you know, they just last summer struck down Vermont's labeling law within a month that it came out. So now no states can require labeling. The executive branch, there might have been a chance under Obama, but we couldn't get through to him. Today, what can you say? The tragedy is that alternative facts are bad enough in the sphere of politics, but they shouldn't be, holding sway in the field of science, and they are when it comes to GMOs and probably too many other issues too. But alternative facts have no place in science, and yet they've been enshrined when it comes to, and they're actually ruling the dialogue in the media and in the scientific community when it comes to GMOs. It's really astounding, but it can be overturned.
Over here.
Your talk focused on risks. I'd like to hear more about observed harms, and I'd like to mention one of them.
Well, you can start off by mentioning it.
The crops that are made to be Roundup resistant get more Roundup, and we get more exposure, and that's
not a good
definite harm.
situation.
Yeah. Anyway.
Yeah. The reason I didn't focus on the Roundup itself is just because, A, most people do, and Roundup is bad, and it's been demonstrated to be a toxin. And of course, it's also, many scientists and scientific organizations now have classed it as a probable human carcinogen as well. But there's research that's demonstrated it's toxic. And that was published in a peer-reviewed journal. Monsanto led a big attack against it, was able to get that research, that study retracted under basically fraudulent pretenses. And I can go into it a little more if you want to. There's a big section in the book, but it's been republished anyway. That was a solid toxicological study. On that basis alone, Roundup should have been off the market a couple years ago. And
Other harms.
but
The Roundup.
let me mention another thing about it. But that same study, it studied the Roundup-ready corn that hadn't been sprayed with Roundup, and Roundup administered in the drinking water without the corn at the level that would have been ingested if the genetically engineered corn had been sprayed, and it was found that both were toxic independently of one another. So that answers your question even more. So even unsprayed, that particular genetically engineered variety of Monsanto's corn was found to be toxic. And the Roundup itself was found to be toxic. And yet, both are still on the market. So, that's the state of, of the science today. And it is important to know also, just on Roundup, even there's a risk of Roundup, even if you're eating, non-GMO wheat or oats, most grains, I think, many legumes, because Roundup for the last several years has been used as a desiccant, pre-harvest desiccant. It's sprayed on the crop shortly before harvest to dry it out, and then I was told too that even after that it can be sprayed again to help in processing. So, but this is just before it's harvested. So there's going to be fairly high, significant levels of Roundup on the grain. So if you're eating bread or other wheat products or oats or rye, my understanding is it's a fairly common practice now to use the Roundup or other form of glyphosate as a toxin, as a desiccant pre-harvest. So it's even more important to be trying to find organic foods because legally they're not supposed to be, sprayed. So this is just another unsettling fact. I'm sorry to be the messenger, but you got to be careful these days if you live in the U.S. It's just the food. They're really messing around with it in ways that are not very friendly.
Okay, all of a sudden I have a lot of hands popping up here. So let's take a few more questions. Yeah, it's almost time to call it. If we can try to keep the answers a little more brief, that's
Okay.
all right. And then we can get maybe 3 or 4 questions in. And then any other people who have questions, we'll just start the break and you can come up and speak with Stephen directly. So here we go.
You seemed a little bit surprised that big money can influence so much, but in fact, that's always been the case, and it will probably continue to be the case for a long time, that a company like Monsanto owns enough senators and congressmen to pass any laws they want, which they have done. So the question is, why are we surprised that that is the case?
Well, I don't know if many people here were surprised, but I said it's surprising how bad science, because, and this is an important point, it's not just Monsanto. In fact, most activists focus on Monsanto, and many people have the idea if Monsanto goes down, then the whole thing will be over. That is not the case. That's why my book doesn't focus on Monsanto much. I focus on the subversion of science and the degradation of science and the lack of integrity that's been displayed by the Royal Society, the National Academy of Science, many eminent university of scientists. And as my book makes the point, they have enabled Monsanto. If they hadn't been misrepresenting the facts and acting as spin doctors, then Monsanto probably would have never made the investment in the first place. And this is a very important point. I'll try to make it quickly. As my book shows, the major misrepresentations were already being made by the scientific establishment, university researchers, and important institutions well before the founding of the very first biotech industry. So the biotech industry was not the first. In fact, biotech industry might not have gotten going if the scientists hadn't been misrepresenting the facts. So the first 2 chapters of my book go into that history. So my book is also a history, and it lays primary responsibility at the feet of the scientists.
Okay, right here.
I try very hard when I bring food to bring non-GMO, and right now on that table there's nothing that's GMO, and
Wonderful.
I try to continue that.
That's great. Congratulations. Thank you.
I know the farmers that grow each and every item on the table.
Okay, terrific. Thanks so much for that.
Thank
Okay.
you.
Will be you, and then
I just ordered Tony Mitra's book. He is a Canadian retired scientist who has compiled 8,000 measurements that the Canadian government does of glyphosate on their foods. And he does it by country of origin. And it turns out that even in organics, I'm so avidly organic, I was so disappointed to see that many of our organics are contaminated. And my theory is, because I come from a farming family, and we also did wholesaling, my family wholesaled yams, that they are probably spraying the organics, say, with, to kill the bacteria, because it's cheap, you know? Yeah, but anyway, Tony Mitra's book, it's horribly expensive. I bought it so to use as a reference for lectures and things, and it's our government doesn't test for it. They're just so remiss, but Canada does, and so he did all the compilations. It's not written. The quality of the writing is putrid, but he didn't have an editor, I don't think. But the data is there. The data is excellent.
Great, thank you. Last one.
Yes, that's kind of a bounce-off question. I'm really thankful to hear that about Canada because I was curious. Glyphosate isn't really new in the terms of things being new nowadays. They have to be like less than a year old to be new. But where can we get information about the glyphosate levels that are detected in us personally by any lab in any region like the Northeast or here or the Northwest or anywhere? And where can we get information about glyphosate levels on produce that is purchased and selected out of Safeway and other places? Like, where do we get that
I'm going to
data?
pass on that and ask if anybody here knows answers. Okay.
The Great Plains lab, I don't remember where they're located, but you can just order the kits online, and you just ship your result, and they will give you the glyphosate result. And then they do organic acid tests too, so they do that.
Great Plains Lab.
Yeah.
All right, great. Please thank Stephen Druker. Yeah, so we have a few copies of his book available for sale at the desk. I don't know how much they are, but $20, okay. So if you want a copy, race to the table.